Hi friends, today we'll be discussing Scoto-Active Stomatal Movement. It's in continuation to the earlier video in which we have already discussed the photoactive stomatal movement. In this Sukhoto-active movement, you will see it is the reverse of photoactive stomatal movement. Here you will see Scoto-active stomatal movement means stomata open at night and close at day. But in photoactive, we have seen that stomata opens during day and closes at night. So stomata open at night and close during day time. Such movements are called as Scoto-active stomatal movements. It's found in succulents as well as in some cam plants. Succulent is a Latin word, succus, means a juice or a sap, meaning a juice or a sap. These plants have a thick fleshy tissues such as leaves and stems which are adapted to store water under semi-arid or arid conditions. They also have reduced stomata. There are some of the examples: Portulacaria, Echeveria, birds or donkey's tail. So these plants have within them an adaptation to store water under arid or semi-arid conditions. In addition to this, some succulents are also cam plants which have a crassulacean acid metabolism pathway. This we will be discussing during photosynthesis. So members of some family, Crassulaceae, Cactaceae, Agaveaceae, Orchidaceae, Liliaceae, they show a certain mechanism, a pathway which is called as crassulacean acid metabolism. They are also growing under arid conditions. Some of the examples are Opuntia, Aloe, Bryophyllum, Begonia, Agave, pineapples, jade plants, and sedum. So these are some of the examples of succulents as well as certain succulents which have the cam type of this metabolism which is called as crassulacean acid metabolism. These are some of the pictures of these plants which I have already mentioned here. These are succulent plants. You'll see the variety of these plants. I have displayed them so that you will be well versed with these types of the plants. They are ornamental plants also. So this is the Echeveria, this is Portulacaria. These are some of the varieties of these sedum plants, and this is called as burro's tail, or it's also called as the donkey's tail. Cam plants which are also succulents, one is the jade plant, this is the pineapple, Opuntia, which is the cactus, and you have agave, you have aloe vera, you have bryophyllum. These are some of the plants which are basically succulents, and they are showing the Scoto-active stomatal movement. Let us discuss the mechanism of Scoto-active movement. As you know, Scoto-active movements, according to Nishida, the stomata is opening during night or darkness, and there is an incomplete oxidation of carbohydrates. There is a low internal carbon dioxide concentration, and during night there will be an accumulation of organic acids, and there is a partial anaerobiosis because there is less oxygen available, and there will be dark acidification. All these things favor stomatal opening in dark. That means during night there is an incomplete oxidation of glucose, carbohydrates. As a result, there is a formation of malic acid that will result basically in the opening of the stomata. So during night, there is an accumulation of acid or acidification in these plants. Because of the partial anaerobiosis, it also produces phosphoenol pyruvate by glycolytic pathway. Because less oxygen is available, that time there will be formation of phosphoenol pyruvate PEP by means of glycolysis, as well as during darkness the stored carbohydrates are also converted into PEP. So that means this phosphoenol pyruvate will combine with carbon dioxide to produce malic acid. So in these plants during night, there is a production of malic acid and it's being catalyzed by PEP carboxylase or PEPCO and malic dehydrogenase. As a result, there is a production of the malate or the malic acid, which is stored in central vacuole of the guard cells. So there can be a situation pyruvate plus carbon dioxide in presence of malic enzyme that can directly form the malate, or there will be an PEP that will combine with the carbon dioxide in presence of an PEP carboxykinase forming an oxaloacetic acid, or the PEP can combine with the carbon dioxide in presence of PEP carboxylase to form oxaloacetic acid, or oxaloacetic acid in presence of a malic dehydrogenase can form the malate. So there will be production of malic acid in the central vacuoles. This malic acid dissociates into protons and malate ions. There will be an efflux of protons and influx of potassium that will take place between guard cells and epidermal cells. As a result, the potassium which enters inside the guard cells will combine with malate to form potassium malate. Potassium malate passes into vacuole to increase its osmotic potential. That is, the cell surface in the vacuole will have an increased osmotic potential. As a result, because of high osmotic potential in guard cell vacuole, it will have a low water potential also. So in relation to epidermal cells, there will be a potential difference. The water will move from the adjacent epidermal cells into the guard cells by endosmosis, and it will favor stomatal opening, as we have seen in case of the modern concept. So it can be explained the epidermal cell and the guard cell. There is a relationship between them. When starch during night is converted into PEP in presence of PEP carboxylase, the carbon dioxide which will be absorbed during night or which is present within the due to partial anaerobiosis, and there will be formation of the malate as well as the protons. Protons will be actively exchanged with the potassium. Potassium will be taken inside. To balance it, the chloride ions will be also taken. As a result, potassium malate will be produced. That will create a osmotic potential here, decrease in the osmotic potential. As a result, the water will move from, and there will be a less water potential here. As there is an increase in osmotic potential, there will be less water potential. Water potential in comparison to this, water will move inside the guard cells. That will result in the stomatal opening. But during daytime now, this is happening during the night. During daytime, there is an availability of oxygen, and the accumulated organic acid which is there, that breaks down rapidly. It undergoes malic acid decarboxylation, or that is called as deacidification. And the carbon dioxide which is released there is utilized in photosynthesis during daytime. It also produces pyruvic acid. Pyruvic acid may be oxidized to carbon dioxide by Krebs cycle and reconverted to PEP or phosphoglyceric acid. PEPCO, which is enzyme during daytime also, which is there as the nighttime also, which is fixing the carbon dioxide, but during daytime it is inactive and it's prone to malate inhibition. When there is a malate, that will inhibit the PEPCO. As a result, what will happen now? There will be PEP or PGA are used for synthesis of sugar by C3 cycle. That means during daytime, there is, instead of a PEPCO, Ribisco is fixing the carbon dioxide now. And there is a carboxylation that results in the formation of the carbohydrate during daytime. So no ion exchange takes place. It leads to the closure of the stomata during daytime. So we have a malic acid during day in presence of oxygen. It will be completely oxidized and it will produce the carbon dioxide. That carbon dioxide will be used in photosynthesis, fixation by means of the C3 cycle. And this is how malate, we have already seen it during daytime. It can be explained with this simple diagram. You have here a stomata, which is during daytime, it's closed, during nighttime, it is open. Now this stomata, during nighttime, what happens there? The glucose in presence of less of the oxygen under anaerobic respiration, there is an accumulation of organic acids. Malic acid is produced there, so there is no released carbon dioxide. As a result, pH of the guard cells becomes high. And at the same time, the carbon dioxide which is available, which is being taken during the night, will be utilized for acidification. That is, this carbon dioxide will combine with the PEP. In presence of an PEPCO, during night, it will form the malic acid. Malic acid will undergo this dissociation into malate and protons. There will be an exchange pump between the epidermal cells and the guard cells. As a result, what will happen? Within these guard cells, there will be an increase in osmotic potential and decrease in water potential vis-a-vis the epidermal cells. There will be an endosmosis, and during that endosmosis, the stomata will open. That means it will be the night from day to night. That means during night stomata opens, and during day it is closed. How it will be closed now during daytime? There is a light falling on this. There is oxygen available. Aerobic respiration will take place, and there will be no accumulation of organic acids because there is complete oxidation of malic acid. So no acid deacidification is taking place there. So no proton, potassium ion transport in guard cells. In addition to this, the carbon dioxide which is released there because of the aerobic respiration, it will be taken by the C3 cycle for fixation to carbohydrates. And also there will be a situation that malic acid which is there will undergo the deacidification, form the PEP, and the carbon dioxide released will be used for C3 cycle. So there will be no ion exchange. Osmotic potential of the guard cells will decrease, and the water potential of the guard cells will increase. As a result, there will be an exosmosis in the guard cells. The water will come out of these guard cells to the epidermal cells, and the stomata shows in closure. This is the simplified this scheme showing the Scoto-Active Stomatal Movement in succulents. What is the significance of Scoto-Active Stomatal Movement? It gives adaptive advantage to plants. It helps to conserve their water content under xeric conditions because it allows them to close the stomata during day and keep them open during the night. It empowers the plants to continue photosynthesis during daytime, even when stomata is closed. It helps plants to efficiently utilize carbon dioxide, absorbed during night and released during respiration. So, direct fixation of carbon dioxide acts as a survival valve to these plants. It is advantageous to succulents, providing ample carbon dioxide for photosynthesis. This is the significance of an Scoto-Active Stomatal Movement for these plants, for these succulents, which are showing this type of the mechanism. What are the evidences in favor of this Scoto-Active Stomatal Movement? Scoto-Active Stomatal movements have been found to be controlled by ion fluxes, as it occurs in other plants, by these scientists. Organic acid produced in night, followed by absorption of potassium from neighboring epidermal cells, leads to opening of stomata. Paul's 1969 and earlier in 1972. Both malate formation and stomata opening in CAM require small concentration of oxygen. Two types of PEPCO have been found in green cells of CAM plants, light form and dark form. Peppo shows diurnal regulation in camp plants. That is, they remain active during night and inactive during day. So that means there are some evidences, direct evidences, which show that the Sukhoto-active movements are being taking place, and they are responsible for conservation strategies in the plants, especially in the camp plants. So this was about the Sokoto-active movements. Now, since we have finished the transpiration, there are certain points which are to be remembered, First is this Kurtz 1928 considered transpiration as a necessary evil or an unavoidable evil for plants. Now you should be in position to answer it because you have understood it, why it is a necessary evil. Diagrammatic representation depicting size of stomata at different times of the day was first prepared by Wani. Stomata are absent or non-functional in submerged hydrophytes. The quantity of water transpired by a unit area of leaf surface per unit time is called as transpirational flux. Quantity of water transpired by a unit area of leaf surface per unit time is called as transpirational flux. Transpiration ratio is the amount of water lost through transpiration per unit of dry matter produced during the growing season of plant. It is around 200 to 300 units for sorghum and 50 for pineapple. Fully opened stomata measures about 3 to 12 microns in width and 10 to 14 microns in length. Number of stomata ranges from 100 to 60,000 per square centimeter of leaf. Number of stomata per unit of leaf is called as stomatal frequency. Salisbury 1927 discovered a correlation between the number of stomata and the epidermal cells per unit area. The percentage number of stomata as compared to all the epidermal cells, including stomata in a unit area of leaf, is called as stomatal index. It's calculated by I S E plus S into 100. I is stomatal index, S is number of stomata per unit area, E is the number of epidermal cells per unit area. Stomatal index is constant for species and has great diagnostic importance. Fresh weight of a plant or a leaf would be maximum in the morning and minimum in the afternoon. Plants growing at high altitudes show xeromorphy, that is adaptation to minimize transpiration. Scoto-active movement and its characteristic of succulent plants can be measured. There are the Scoto-active movements and its the characteristics of a succulent plants. As we have seen, these movements are characteristic of succulent as well as camp plants. So transpiration can be measured with the help of certain instruments, measuring instruments. They are called as potometers. They are used to measure the transpiration, and potometers are based on the assumption that rate of transpiration is roughly equal to the rate of water absorption. Different types of potometers are Farmer's potometer, Genong's and Bohr's potometer. Carbon dioxide gas also works as an anti-transparent. Blue light of visible spectrum promotes stomatal opening, and generally stomata are photoactive, that is, they open during day and close at night. These are some of the points which are to be remembered, because we have concluded the mechanism and the major portion of the transpiration. So in this video, we have seen that what are the Scoto-Active Stomatal movements, how they are taking place, how there is an acidification and deacidification, acidification during the night and deacidification during the day, and how stomata opens and what is the significance of that opening during the night and during the closing during the day. I hope you have understood this mechanism, and in future videos we'll be discussing the antitranspirants and the factors affecting the transpiration. I hope you have understood in detail now the various mechanisms, both photoactive and Scoto-active stomatal movements. With this, thank you for the patient listening, and till the next video, be safe and be happy. Thank you very much.